Practical 4, Cell Structure, Group 4, prepared by Muhammad Aiman Haikal bin Abdul Halim, Nur Amira binti Yahya, Suhaila binti Ahalin, Ain Hanani binti Azrur. Introduction. Cell is a basic life structure which is functioning in one organism. This basic unit conducting processes that enable one organism survives and functions. A typical cell consists of a cell membrane or plasma membrane and protoplasm. Protoplasm is bounded with cell membrane and consists of a nucleus which is surrounded with complex cytoplasm. Cytoplasm contains organelles such as Golgi apparatus, rough and smooth endoplasmic reticulum, ribosome, mitochondria, lysosome, vacuole, and chloroplast. Objectives of this practical is to learn and to identify the organelles and cell structure, to identify the differences between animal and plant cells, and to learn slide preparing techniques.
The materials and equipments that we use for the experiment is distilled water, iodine solution, slide and cover slip, wet tile, onion, forceps, and bleed. Using a sharp knife, peel a small portion of the epidermis layer of the onion. Next, place the epidermis layer on a slide which has been dropped with 2 to 3 drops of water. Then, gently place the cover slip on top of it. After that, observe the slide under the microscope with low and high magnification power. Then, drop 1 or 2 drops of iodine solution at one end of the slide. The solution will spread under the cover slip when a filter paper is placed on the opposite end. Lastly, observe the slide under the microscope to identify the cell wall, nucleus, vacuole, and cytoplasm. So these are the results after we observe the slide under the microscope, so we can identify the cell wall, nucleus, vacuole, and cytoplasm of the onion epidermal cell under 40 times magnification.
First, place a piece of elodea on a slide, which has been dropped with a drop of water. Then, cover with a cover slip. Observe the cell under low and moderate power. Observe the moving green dots. The green dots are called chloroplasts and important in photosynthesis process.
And this is the structure of the elodea leaf cell.
These are the materials and equipments needed. Using a toothpick, gently scrape the inner layer of your mouth. Drop sodium chloride 0.9% solution on a slide. Then, place the cells on the sodium chloride 0.9% solution. After that, color the cell with methyl blue coloring and cover with the cover slip. Use filter paper to get rid of excess solution. Lastly, observe the cells under low and moderate magnification power. And this is the structure of chick cell.
The last experiment, we are using the reptile blood slide. Observe the prepared slide by using a microscope, and the picture shows the result from the slide observation of reptile blood by using a microscope.
An onion is a multicellular which consists of many cells plant organism. As in all plant cells, the cell of an onion peel consists of a cell wall, cell membrane, cytoplasm, nucleus, and a large vacuole. The nucleus is present at the periphery of the cytoplasm. The vacuole is prominent and present at the center of the cell. It is surrounded by cytoplasm. The presence of a cell wall and a large vacuole are indicators that help to identify plant cells such as seen in the onion peel. Then, no chloroplast can be observed in the epidermal cells of onions because epidermal cells do not carry out photosynthesis.
Each allular leaf cell exemplifies a typical plant cell. It has a nucleus and a stiff cell wall which gives the cell its box-like shape. The numerous green chloroplasts allow the cell to make its own food by photosynthesis. The central vacuole takes up most of the volume of the cell. It is transparent, but we can see where it's pressing the chloroplast up against the cell wall, especially at the end of the cell. Like animal cell, the cytoplasm of the plant cell is bordered by a cell membrane. The membrane is so thin and transparent, but we cannot see it. But it is pressed against the inside of the cell wall.
This human cheek cell is a good example of a typical animal cell. It has a prominent nucleus and a flexible cell membrane, which gives the cell its irregular, soft-looking shape. Like most eukaryotic cells, this cell is very large compared to prokaryotic cells. For scale, notice the pair of dark blue bacterial cells sticking to the right edge of the cheek cell. The bacteria are only a fraction of the size of the nucleus, but their tiny size is typical for bacteria.
Discussion. Erythrocytes of reptiles are ellipsoidal and have a centrally positioned nucleus. Depending on the state used, the color of the cytoplasm ranges from orange to pink or pale violet. Red blood cells are typical for reptiles. They are large with irregular nuclei with abundant cytoplasm. The cytoplasm frequently contains some blue indistinct granules, which are more probably degenerate organelles accumulating with time. In conclusion, we can conclude that plant and animal cells differ significantly. Plant and animal cell maintains its constant internal environment, homeostasis, in which the structure of each cell depends. Here are a few of the differences of plant and animal cells. Cell membrane. Plant cells is surrounded by a rigid cell wall, while the cell membrane of the animal cells is not surrounded by a rigid cell wall. Plastids and chloroplasts. Plant cells have these, but animal cells do not have this. Shape. The plant cells are larger with regular shapes, while animal cells viewed under a microscope appear smaller and irregular in shape. Plant cells contain a large central vacuole, while vacuoles in animal cells are less in numbers and size.